Good morning, everyone, and welcome following the introductory address from our president. It's a great pleasure to begin the session on ion channels in pancreatic physiology and pathophysiology. I'm Robert Sutton, and together with my co-chairs, Fred Gorlick from Yale and Ole Peterson from Cardiff, it's a great pleasure. Ion channels are proving to be a vital component of pancreatic disease, and now not just the basic and translational research, but clinical trials are underway across pancreatic diseases. So it's a really pertinent topic for us to learn about. Our first speaker is a representative of the Hungarian groups that have been working on cystic fibrosis transmembrane conductance regulator ion channel for a long time and advancing our work in this area. And it's a great pleasure to introduce one of the leaders, Victoria Vengelovec, who will talk about CFTR restoration in acute pancreatitis. Over to you, Victoria.
Dear chairs, dear colleagues, in my presentation, I'm going to talking about the beneficial effect of CFTR restoration in acute pancreatitis. So acute pancreatitis is an inflammatory disease that requires short or long-term hospitalization in most of the cases. The incidence of acute pancreatitis has been increased in the last few years, placing a heavy burden on the healthcare systems. The major etiological factors in the development of pancreatitis are heavy alcohol consumption and gallstones, which are responsible for more than half of the cases. Most of the patients with acute pancreatitis show mild symptoms. However, in approximately 20% of the patients develop severe or moderately severe pancreatitis, and in the severe form, the mortality rate can reach almost 30%. Although the therapy of acute pancreatitis has been changed and improved a lot in the last few years, there is still no specific therapy. Therefore, the identification of novel drug attack point is crucially important. In order to find a drug attack point, it's important to understand how the exocrine pancreas works. Basically, there are two major cell types in the exocrine pancreas, the acinar cells and the ductal cells. The acinar cells secretes more than 300 bioactive molecules and a significant amount of hydrogen ion, resulting an acidic pH of around the acini. In this acidic pH, the trypsinogen autoactivation can occur more rapidly, which can lead to pancreatitis. There is a protective mechanism which can act against this acidification, namely the ductal bicarbonate secretion. Therefore, the physiological function of the ductal cells is extremely important. On this schematic picture, you can see pancreatic ductal epithelial cells. As you can see, many transporters play a role in the ductal bicarbonate secretion, and among them, I would like to highlight the role of the CFTR chloride channel. The CFTR is a cyclic EMP activated chloride channel, which regulate the transepithelial fluid and salt transport. In most of the epithelia, the CFTR is localized to the apical membrane, and in the ductal cells, together with the chloride bicarbonate exchanger, play an essential role in the secretion of high bicarbonate fluid. In the case of improper function of the CFTR chloride channel, the fluid and bicarbonate secretion is decreased, and this can increase the risk of cystic fibrosis or the development of pancreatitis. It is well known that the different CFTR gene mutations in the cystic fibrosis patients can increase the risk of pancreatitis development. However, the role of CFTR in the pathomechanism of pancreatitis inducing factors is not well known. One of the significant findings of our work group is that alcohol, which is one of the major etiological factors in pancreatitis, so alcohol decreases the expression and activity of the CFTR chloride channel, which leads to decrease the bicarbonate secretion, and as a result, acidosis and hypovolemia develops. In this acidic environment, as I previously mentioned, the autoactivation of trypsinogen accelerates, which can lead or aggravates pancreatitis. So based on these results, we hypothesize that restoration of the function of the CFTR channel can be beneficial in acute pancreatitis. There is a clinically available and approved drug, the Orkambi, which has been recently developed. And this drug is used effectively in the therapy of cystic fibrosis patients. The Orkambi contains an activator, the ivacaftor, and the corrector, the lumacaftor. And these two drugs can improve the function of the CFTR channel in different loss of function mutations. So based on the beneficial effects of Orkambi on the CFTR channel, we wondered if this drug is able to restore the CFTR damage caused by the ethanol. And if so, our second question was whether this drug can improve the course of pancreatitis. Therefore, our aim in this study was to test the active substances of the Orkambi in the alcohol-induced CFTR damage. So intra-interlobular ducts were isolated from the guinea pig pancreas, and the CFTR expression was investigated by immunohistochemistry. The ducts, before the immunohistochemistry, the ducts were pretreated with ethanol and the activator and corrector, and here you can see the experimental protocol. So the ducts were pretreated with different concentration of ethanol for 12 hours, and the effect of the activator and the corrector were investigated at different time points. On this representative picture, you can see that ethanol dose-dependently decreased the expression of the CFTR chloride channel. We separately investigated the expression of the CFTR in the apical membrane and in the cytoplasm. And we found that in the apical membrane, the ethanol dose-dependently decreased the expression, whereas in the cytoplasm, the expression of ethanol, the expression of CFTR only decreased at higher concentration of ethanol. Next, we investigated the effect of the activator and the corrector. Both drugs on their own had no effect on the expression of CFTR, neither in the apical membrane nor in the cytoplasm. However, the corrector was able to dose-dependently increase the expression of CFTR after the ethanol treatment. Similar results were found with the activator, which also dose-dependently increased the expression of CFTR after 30 millimolar ethanol treatment. And in both cases, both in the case of the corrector and in the case of activator, the 10 micromolar concentration was the most effective. So next, we investigated the combination of the two drugs, and the combination was also effectively prevented the inhibitory effect of 30 millimolar ethanol, and the effect of the combination treatment was higher than the effect of the VX770 or the VX809 alone. So in the combination treatment, also the 10 micromolar concentration was the most effective. Therefore, we use this concentration in the further experiment. So next, we test the effect of the combination at different concentrations of ethanol. And we found that the combination treatment was able to prevent the inhibitory effect of 30 and 50 millimolar ethanol, but it was ineffective in the case of 100 millimolar ethanol. So in the following, we also investigated the activity of the CFTR channel. For this, we isolated individual ductal cells from the intact ducts, and using the whole cell configuration of the patch clamp technique, we measured the activity of the CFTR chloride channel. So the activity of the channel is relatively low under resting condition. Therefore, we measured the activity under forced choline stimulated condition. So we found that 30 millimolar ethanol strongly decreased the activity of the channel. However, both the activator and the corrector were able to prevent the inhibitory effect of ethanol. And when the two drugs were added together, their effect was additive. In order to investigate the in vivo effect of Orkambi, we induced acute pancreatitis in the guinea pig by cerulane. And after the third cerulane injection, the Orkambi was administered per dose in a single dose. And then histological and laboratory parameters were investigated. On these representative pictures, you can see that the cerulane induced the infiltration of leukocytes, necrosis, and edema. However, those animals which were orally treated with Orkambi, the severity of pancreatitis significantly decreased. The beneficial effect of Orkambi was also observed in the laboratory parameters. So both in the serum amylase level and the rate of necrosis significantly decreased due to the Orkambi treatment. So in conclusion, we found that Orkambi is able to restore the alcohol-induced decrease in the CFTR expression and activity. And we also found that Orkambi reduces the severity of experimental pancreatitis. These taken together, these results indicate that restoration of the CFTR function can be beneficial not only in the cystic fibrosis, but also in acute alcohol-induced acute pancreatitis. Thank you for your attention, and I'm happy to answer your questions. Thank you.
Thank you very much, Victoria. I hope that you can hear me. Thank you. We have 5 minutes for questions. We have to keep strictly to time because there's a session that comes on after us, and we have to be fair to all the participants, 20 minutes each, including the questions. So one of the things that has already appeared in the chat is a comment from Maria Lourdes Ruiz Ribolo, who says it's an great talk, nice talk, and not difficult to be understood by a clinical pancreatologist, for which we thank you very much. And another comment is some of the idiopathic recurrent acute pancreatitis patients have single nucleotide mutations in CFTR, which apparently are not significant clinically, but this may hinder acute pancreatitis. That's the comment. Mm-hmm. Will it be the solution for them? So for whom will lumacaftor and ivacaftor, the combination working in different ways, most be appropriate, do you think? How would you take this into the clinical arena if you think you have sufficient evidence to do so?
Thank you for these questions. So basically, the Orkambi was the most effective in patients with homozygous F808 mutations. So in this case, due to the abnormal post-translational modifications, the protein is unable to localize to the apical membrane. So based on our result, we hypothesized that it can prevent the recurrent acute pancreatitis in patients with alcoholic pancreatitis. But there is not too much. The effect of Orkambi has been only investigated in one cases. So in this case, the cystic fibrosis patients was treated with Orkambi. And in this case, the exocrine function of the patients improved 3 years after the Orkambi treatment. But there was no other study in which the Orkambi was investigated in acute pancreatitis.
Okay. There are quite a lot of questions appeared, and we're not going to be able to take them all because we've only got 2 minutes left. So Fred has asked, Fred Gorlick, who I have to give enormous credit to for getting up in the really small hours of where he is. I don't know what time he rose, 2 a.m. maybe. So he's obviously a very astute man. What effects does CFTR inhibition have on duct cells in the context of acute pancreatitis? Is there inflammasome activation?
Yes, so inhibition of the CFTR chloride channel will decrease because the chloride channel works together with the chloride bicarbonate exchanger. This will decrease the bicarbonate secretion, and as a result, this will lead to decrease the fluid secretion. And the decreased fluid secretion can increase the risk of inflammatory processes, and this can increase the risk of pancreatitis development.
Okay, fair enough.
This is the link between the image of the CFTR chloride channel and the development of pancreatitis. So it can promote the premature activation of digestive enzymes in a low bicarbonate environment.
Okay. Well, there are further questions, including from Bill Greenhalgh and Minotti Apti, but it is the 20 minutes are up, and I would encourage them to communicate with you directly. I'm sure that would be appropriate, either through the chat function in the meeting or subsequently or via email or however, but it's clearly a very important item of work and potentially extremely promising. So thank you so much, Victoria. That's really great.
Thank you very much. Thank you.
So the next speaker will be introduced by Professor Fred Gorlick from Yale. He's having difficulty with his internet stability, but I hope he will be able to do this. If not, we can rapidly transfer arrangements. So Fred, are you able to?
I'm able to at the moment. Robert, can you all hear me?
Yes, thank you. That's great.
Okay. Well, again, I'd like to thank the organizers and especially Dr. Lohr for inviting me to participate. I had hoped to be there in person to visit the city where my mother was actually born, and hopefully that will happen at some time in the future. near future. So it gives me a great pleasure to introduce Julia Gerasimenko. Julia is on the faculty at the Cardiff School of Biosciences in the UK. I think notably she completed her PhD at the Bogomolets Institute of Physiology in Kiev, Ukraine. I think we're all very familiar with, those of us in science are very familiar with Julia's work. She's really done some of the pioneering work on pancreatitis injury in the acinar cell and the mechanisms, particularly as related to zymogen activation and calcium signaling. And in recent years, she's extended her expertise to the role of other pancreatic cells in the pancreatitis response, including immune cells and stellate cells. So she's been giving an ambitious title, which is the role of different cell types in exocrine pancreas in health and disease. I'm looking forward to hearing her presentation and how she addresses this important topic. Julia?
First of all, I would like to thank the organizers of the meeting for inviting me to speak today. And also, I like to show my information about myself. So I'm PhD senior lecturer at Cardiff School of Biosciences, Cardiff University, Wales, United Kingdom. And I do not have any relevant financial relationship to disclose. Today, I'm going to present our recent data and discuss the role of different cell types in exocrine pancreas in health and disease. It has been established previously that in exocrine pancreas, pancreatic acinar cells not only dominate the tissue, but also become the origin of main disorders such as acute chronic pancreatitis and pancreatic ductal adenocarcinoma. But there are other important cell types present in the same tissue, such as ductal cells, pancreatic stellate cells, neurons, and also cells of innate immunity, and they all play a significant role in organ function under physiological and pathological conditions. So therefore, I believe it's very important to study ways of communication of different cell types in health and disease. Recently, we have developed technique to isolate and experiment with ex vivo pancreatic lobules that allows us to study different cell types in their natural environment. We can visualize pancreatic acinar cells and stellate cells, which you can see located in the periacinar space with the help of Q4 staining of the lobule. We can also identify different cell types. You can see this experiment here. Pharmacologically, pancreatic neurons, calcium signals can be observed so when we depolarize membrane with a high potassium solution, and the nerve cells is actually color-coded here in purple. Acinar cells color-coded in blue, which are non-excitable cells, usually respond with a delay to depolarization as a result of action of neurotransmitters released by the neighboring neurons. Stellate cells respond to blood pressure lowering nonapeptide bradykinin, and immune cells and some acinar cells respond to ATP ligation. It also has been established that acute pancreatitis-inducing agents such as bile and alcohol induce calcium signaling abnormalities that are responsible for the disease development when pancreas starts to misbehave. These abnormalities include intracellular calcium overload in pancreatic acinar cells due to massive calcium release from the internal stores, followed by the excessive calcium entry through the store-operated calcium entry channels, such as calcium release activated calcium crack channels. This calcium overload triggers the premature calcium-dependent intracellular activation of digestive enzymes such as trypsinogen to trypsin, mitochondrial calcium overload, and dysfunction of this organelle, unable to produce ATP, so the ATP level in the cells drops. And so all this leads to the necrotic cell death and inflammation of the tissue. About a decade ago, we proposed that to crack pancreatitis by inhibition of crack PORAI1 channels to protect pancreatic isthmus cells against the acute pancreatitis injury. A crack channel inhibitor produced by GlaxoSmithKline GSK 79758 was very effective to protect pancreatic isthmus cells against the protease activation and cell necrosis in vitro studies, which makes a potential tool in antipancreatitis therapy. Our results then later were elegantly confirmed by Robert Sutton group. They provided the evidences from in vivo studies. Currently, a novel small molecule PORAI1 inhibitor, which is selective crack channel inhibitor, CM4620, with the commercial name Xora, developed by Calcimedica, has completed the phase 2 clinical trial for the treatment of severe acute pancreatitis and for the patients with acute pancreatitis side effects from pediatric acute lymphoblastic leukemia treatment with asparaginase, and also, as you can see, COVID-19 pneumonia. However, we must understand the long treatment with this inhibitor for acute pancreatitis. So it's questionable due to profound effect on immune cells. We recently discovered that pancreatitis-inducing agents such as alcohol, bile, asparaginase inhibit hexokinases, enzymes responsible for the conversion of glucose to glucose 6-phosphate as an important initial step of glycolysis. Galactose, however, does not require hexokinases activity to keep glycolysis running and to maintain the ATP production. So we have demonstrated that supplementation of drinking water with galactose in vivo experimental mouse model of acute pancreatitis markedly reduced the main hallmarks of the disease, including the necrosis and inflammation, and the total histological score was dramatically reduced. So it makes this galactose as a very attractive, promising, and simple way of treatment of this disease. So what we know about pancreatic stellate cells? They play a pivotal role in pathological fibrosis of pancreas during chronic pancreatitis and pancreatic cancer. So usually they are in fasting state, and they perform housekeeping role by storing vitamin A, and it's shown here with the help of multi-photon imaging, so the vitamin A droplets. Bradykinin concentrations, high bradykinin concentrations usually usually induce the calcium signals in stellate cells. And it's also known that during pancreatic injury, these cells become activated. They start to release extracellular matrix proteins, cytokines, and growth factors. The bradykinin is known to be involved in various physiological and pathological processes, but tissue and plasma bradykinin concentrations are very important and are tightly regulated by the body, and they cap at very low levels, about 40-70 picomolar. Interesting that we demonstrated the threshold for activation of normal stellate cells in pancreatic tubule is about 100 picomolar, which is very close to the borderline of normal plasma level concentration of the substance. Any increase in plasma tissue bradykinin level, which occurs under several conditions, including acute pancreatitis, would elicit calcium signals in stellate cells, and from this particular moment in time, these cells become activated and then become dangerous. Previously, we have demonstrated that bradykinin responses in stellate cells are due to activation of B2 receptor. Interestingly, then, when we block beta receptor in stellate cells, we reduce the pancreatic acinar cells necrosis produced by the alcohol or bile. There is some way of communication between these two cell types. However, we propose that the nitric oxide could be the most possible way of the communication. So nitric oxide is produced by stellate cells and can be really diffused to the neighboring pancreatic acinar cells. And just this is the way of cell communication during the disease state. So we have performed an in vivo experimental model of alcohol-induced pancreatitis in mice and followed the functional changes of stellate cells. 48 hours after the induction of AP, pancreatic stellate cells lost their sensitivity to bradykinin in small doses, but instead they gained the sensitivity responsiveness to B1 receptor agonists, meaning that the B1 receptor is already upregulated at this stage. Also, stellate cells gain responsiveness to trypsin and thrombin, indicating overexpression of protease-activated receptor 2. Activation of PAR2 has been implicated in promoting TGF-1-induced cell migration and invasion in pancreatic ductal adenocarcinoma. So that would be a link between acute pancreatitis and pancreatic ductal adenocarcinoma development. For the first time, we were able to follow the functional changes in immune cells, such as macrophages, in live pancreatic globules. So after induction of acute pancreatitis with alcohol injections, so macrophages become activated upon application of immunoglobulin G. And the frequency of calcium signals in macrophages is drastically increased as compared to the control. So after each experiment, we were just confirming the location of macrophages with the help of co-staining of cells with specific fluorescently labeled antibodies, and also nuclear staining. A nucleus of macrophages has a peculiar E-shape, so that could be also recognized. So with the help of immunostaining, we can calculate the density of immune cells in globules, which is significantly increased 48 hours after induction of acute pancreatitis, and also continued to increase on the third day after the disease was initiated. Recently, we started to be interested in the effect of SARS-CoV-2 virus on pancreas. This virus is known to trigger many inflammatory diseases, with several reported cases of associated acute pancreatitis. The virus enters the cell via receptor-mediated endocytosis using binding of S protein to the S1 protein subunit to the ACE2 receptors. ACE2 receptors have been shown to be present in pancreas, but the mechanism and cell type targeted by the virus is still unknown. Taking advantage of this, our novel method of pancreatic globule preparation, so we have demonstrated for the first time that S1 protein at 200 nanomolar typically elicits the calcium signals in spikes or trains of spikes in pancreatic macrophages and also stellate cells. We were also able to block the calcium responses to S1 with the help of Oxora, selective RI1 crack channel inhibitor. What is very interesting and intriguing was to observe substantial delay in development of S1 calcium signals in macrophages that was up to 4 times longer than in stellate cells. We were interested to investigate this fact further, and it has been shown previously that S1 induces release of cytokines, including IL-18. So the incubation of globules with the interleukin-18 binding protein selectively abolished calcium signals in macrophages, but as you can see, responses in stellate cells still were unchanged. Interesting that we did not observe any calcium signals to S1 in pancreatic acinar cells during our experiments. So in the frame of our experiments, we didn't see any calcium responses from pancreatic acinar cells. So we can conclude that stellate cells appear to be a primary target for S1, releasing interleukin 18 and activating macrophages. So the blockage of EL18 with the binding protein selectively eliminates calcium signals in macrophages. The blockage of calcium entry with Oxora suppresses calcium signals elicited by S1 in both cell types, which makes the array crack channels very attractive potential therapeutic target for treating not only inflammation, but also reducing the risk of acute pancreatitis in patients with COVID-19. To summarize, just in conclusion, so abnormal calcium signals in pancreatic acinar cells, stellate cells, and immune cells amplify the pancreatic injury. So the necrosis of acinar cells induced by alcohol, bile, and drugs such as asparaginase causes the leakage of active enzymes, trypsin, kallikrein. Kallikrein supports the production of bradykinin. Bradykinin activates stellate cells, and they produce nitric oxide, which again exacerbates the necrosis process in pancreatic acinar cells, neighboring cells. ATP and ADP activate the resident macrophages. They start to produce cytokines, attract other immune cells to the site of the injury. So injury, inflammation in the site increases the at least twofold the risk of progression from acute pancreatitis to pancreatic ductal adenocarcinoma. I would like to thank my collaborators from Cardiff University, Bogomolets Institute in Kiev, Jena University in China, and also Great Ormond Street Hospital, London, in UK. And also I would like to thank my grant body, Children with Cancer UK. Thank you very much for your attention.
I'd like to thank Dr. Gerasimenko for that wonderful presentation, and congratulations, Julia, you really did put it together. I think it's a very important new concept in pancreatology and pancreatitis, and that is that the response, the cells of the pancreas really act as a coordinated syncytium to modulate disease, and that they're actually, it provides with a number of different points of potential interventions. I would invite our participants to put questions into the chat. First, maybe I could start with a question, Julia, and one that I was thinking about. It looked like on your first slide showing calcium signaling, there was an islet sitting in the midst of the other cell types. And Jason Bruce, I think, and his group has really put forth the concept that insulin signaling and modulation of acinar cell calcium is important in pancreatitis responses. I wondered if you had an opportunity to look at islet calcium responses in the context of pancreatitis.
Thank you very much, Brad. It's a very interesting question. So we haven't looked at the function of islets yet, of course, but this is very a great opportunity, because in our lobules, as you mentioned, so we do have like everything. This is a piece of tissue in their natural environment, so we try to keep it as much as possible intact. And so definitely that would be a very interesting project to look at the function of islets. However, we are mostly in our experiments so far focused on the exocrine parts, and so how it's really so the initial stages of the pancreatitis, which involve only the exocrine first, the pancreatic acinar cells, so how they could, by bursting, could really affect other cells around. And so this is by the normal, well-established causes of acute pancreatitis, such as alcohol or bile acids, etc. However, the new area was for us to investigate the SARS-CoV-2 S1 protein, because that's something also new to us. But yes, that would be nice to also look at the endocrine pancreas as well.
No, I agree. There are many new and novel concepts provided by your presentation. It's going to give us all a lot to think about. The next question comes from Professor Sutton, and he was wondering about the specific responses of pancreatic macrophages in the context of Matthias Sendler's work, showing that macrophages are capable of activating trypsinogen, and that activation can stimulate inflammatory responses in the macrophages. And your thoughts about how this coordinated calcium signaling might fit with Dr. Sendler's data?
Thank you very much. Very interesting question. So, of course, there are many possibilities. So how cells communicate indeed. So we just only started to look at the sequence, so how they might communicate. And the first phenomenon we found, as I explained, with S1 applications, so how macrophages are activated by perhaps stellate cells through. So there are some kind of intermediate communication between cells. Indeed, we know that stellate cells are activated by trypsin, by thrombin, so they could be really on the way to start the process with macrophages. However, again, so it's a very interesting subject for us to look at. And yeah, thank you very much. We need to really to rethink many aspects of this particular question. So just to say, so we can observe only the resident macrophages in the intact tissue, right? So far we were looking at the resident macrophages. However, when we induce acute pancreatitis, so we can also look at the more general presence of macrophages when should arrive, for instance, to the tissue after the injury.
So a question from clinicians attending. The ORA1 inhibitors are in clinical trials. They've advanced. I wondered if you anticipated any downsides from their use, vis-a-vis you're inhibiting some acute inflammatory responses that might be important for suppressing infection. Are there any concerns that you've thought about in this concept?
Well, so to our knowledge, of course, the effect of CM compounds, so it's quite sufficient for to suppress immune cells. However, in a long-term application, that would be quite dangerous, isn't it? So for the severe cases of acute pancreatitis, same as severe pneumonia or other severe symptoms of inflammation, that would be really advantageous. However, when we talk about treatment of, for instance, the leukemia treatment for children, so that could be taken with caution, because when children accept the treatment against leukemia, it's usually long treatment. And if during this treatment, this particular substance would be constantly present, so that long-term suppression of immune system, I think that would be problematic.
I think we should probably draw to a close because we've gone a little bit over time. Fred, perhaps you'd hand over to Ole.
Yes, so on behalf of the moderators, I'd like to thank Dr. Gerasimenko for really a wonderful overview. And I think Julia to be congratulated for bringing a new concept of the integrated response to pancreatitis, to pancreatology. Thank you again.
Well, it's my pleasure to take over now. Greetings to everybody, and particularly to our friends and colleagues in Ukraine. Our thoughts are very much with you, and I send you greetings also from the Royal Society in London, where I currently am. So without any further ado, I'd like to introduce Dr. Joseph Mallet from Szeged University in Hungary, who will talk to us about membrane calcium channels in the physiology and pathology of the exocrine pancreas. So, Joseph, the floor is yours.
Dear chairs, dear colleagues, first of all, I would like to thank the opportunity to present our data for the organizers, and I also would like to thank to organize the European Pancreatic Club meeting. These are my details in a nutshell, and these are my financial disclosures. Epithelial cells play a major role in the physiology and pathophysiology of different organs, such as pancreas, liver, or lung. Epithelial cells secrete bicarbonate-rich fluid, which sets the intraluminal pH in the organs in an alkalic range. Under pathophysiological conditions, this bicarbonate and fluid secretion significantly impairs, which will lead to different pathological conditions such as acute pancreatitis, liver, or lung diseases. In acute pancreatitis, the major etiopathogenetic factors are biliary stone formation and heavy alcohol consumption. And also, we have to know that the mortality of acute pancreatitis is still remained very high. Therefore, we need to identify novel drug targets which can be used to treat these patients. Pancreatic ductal epithelial cells play a major role in acute pancreatitis, and due to the research work of Peter Hedges' group, we have now a better understanding how ductal epithelial cells contribute to this pathological condition. So both bile acids and alcohol can generate sustained intracellular calcium elevation, which will lead to mitochondrial damage and impair bicarbonate and fluid secretion of ductal epithelial cells, leading to acidic intraluminal pH, which in turn enhance trypsinogen autoactivation and promote the development of acute pancreatitis. The source of the sustained intracellular calcium elevation could be a process called store-operated calcium entry. In this process, STIM1 and ORI1 proteins play a major role. STIM1 is a calcium sensor which is expressed in the endoplasmic reticulum, and the depletion of endoplasmic reticulum calcium stores will lead to the activation of STIM1, which will activate ORI1 calcium channels in the plasma membrane, leading to sustained intracellular calcium elevation, due to the influx of extracellular calcium. And due to the elegant work of Philip Peterson's and Robert Sutton's group, we know that the inhibition of ORI1 calcium channel protects pancreatic acinar cells from sustained intracellular calcium elevation and necrosis. And we also know that the inhibition of ORI1 calcium channel in vivo will impair tissue damage in different forms of acute pancreatitis. So ORI1 could be a potential drug target, and there are clinical trials now ongoing, organized by Calcimedica, to assess the beneficial effect of ORI1 inhibition in acute pancreatitis patients. However, we have no information what ORI1, what physiological and pathophysiological roles ORI1 have in pancreatic ductal epithelial cells. Therefore, we used transcriptome analysis in human and mouse pancreatic organoids, and we showed that ORI1 and STIM1 are expressed in the pancreatic ductal epithelial cells, and using immunofluorescence staining, we also demonstrated that ORI1 is expressed on the apical plasma membrane of pancreatic ductal epithelial cells. With functional measurements, we demonstrated that ORI1 is surprisingly constitutively active in pancreatic ductal epithelial cells. When we administrate ORI1 inhibitor without any prior stored ablation, we see a drop in the intracellular calcium level, suggesting that ORI1-mediated calcium influx play a major role under basal conditions in the calcium homeostasis of ductal epithelial cells. Using siRNA-mediated knockdown of ORI1 and STIM1, we demonstrated that this constitutive calcium influx is abolished. On the other hand, the resting intracellular calcium level of the pancreatic ductal epithelial cells significantly decreased, further confirming our observation. Next, we wanted to assess the physiological impact of this sustained extracellular calcium influx in ductal epithelial cells. And thus, the major function of ductal epithelial cells is to secrete bicarbonate and fluids. We investigated the effect of ORI1-mediated calcium influx on the activity of CFTR. So first, we demonstrated that ORI1 and CFTR colocalizes on the apical plasma membrane of the ductal epithelial cells. And we also demonstrated with super resolution microscopy that these two proteins are actually in a very close proximity to each other. They sit within a couple of nanometers. Using functional analysis, we demonstrated that that inhibition of ORI1 with a selective ORI1 inhibitor significantly decreases the CFTR-mediated chloride secretion, suggesting that the constitutive calcium, extracellular calcium influx via ORI1 has a major role in regulating CFTR activity in the ductal epithelial cells. Next, we wanted to assess whether this regulatory interaction is exclusively present in the pancreatic tissue, or it can be identified in other secretory epithelial cells. So we generated organoids from the airway, liver, and also from human pancreatic tissue. And we demonstrated that CFTR and ORI1 colocalizes on the apical plasma membrane, similarly to the mouse pancreatic ductal epithelial cells. And with functional analysis, we demonstrated the same phenomenons. So ORI1 was constitutively active in these epithelial cells, and the inhibition of ORI1 significantly impaired the CFTR-mediated chloride secretion. Next, we wanted to characterize this regulatory nanodomain further, and we assessed the role of adenyl cyclases in this regulatory process. Using transcriptome analysis, we demonstrated that in ductal epithelial cells, adenyl cyclase 3, 6, 8, and 9 are expressed, and also expression of 1 was found on a very moderate level. And with super resolution microscope, we demonstrated that all adenyl cyclases can colocalize with CFTR. However, when we quantified the number of colocalizing clusters, we found that adenyl cyclase 3 has a very moderate colocalization, whereas adenyl cyclase 1, 3, and 8 have high colocalization frequency with CFTR. When we used the siRNA to knock down adenyl cyclase 1, 3, and 8, we found that this knockdown eventually significantly decreases CFTR-mediated chloride secretion in pancreatic ductal cells. To demonstrate to demonstrate this regulatory nanodomain further, we use tricolor D-storm microscopy, and we showed that ORI1, adenylyl cyclases, and CFTR are actually located in the same nanodomain. So to summarize this functional regulatory nanodomain, we can say that in secretory epithelial cells on the apical plasma membrane, ORI1, adenylyl cyclases, and CFTR are localized to the same nanodomain in the apical plasma membrane, and the constitutive calcium influx via ORI1 can enhance the activity of the calcium activated adenylyl cyclase 1, 3, and 8, which can generate high cyclic AMP nanodomain to promote CFTR activity in these epithelial cells and CFTR mediated ion secretion. In the next step, we also wanted to assess the role of ORI1 in different pathophysiological conditions. In the first series of experiments, we investigated the role of ORI1 mediated calcium influx in acute pancreatitis, and using a selective ORI1 inhibitor, we were able to demonstrate that ORI1 mediated calcium influx plays an important role both in the bile acid and alcohol and fatty acid mediated sustained intracellular calcium elevation in the pancreatic ductal cells. And when we investigated the effect of ORI1 inhibition on the function of the pancreatic ductal epithelial cells, we were able to show that inhibition of ORI1 mediated calcium influx is significantly increases the pancreatic ductal bicarbonate secretion and also the CFTR mediated chloride secretion in the presence of bile acids or fatty acid or ethanol and fatty acids, suggesting that the inhibition of ORI1 channel is sufficient to maintain the pancreatic ductal function in the presence of these toxins. We also provided evidence using in vivo models, and first we demonstrated that ORI1 inhibition significantly decreases the pancreatic damage during using different experimental acute pancreatitis models, and we also demonstrated that inhibition of ORI1 channel in vivo can protect the in vivo fluid secretion, in vivo pancreatic fluid secretion in mice, further confirming that ORI1 is a potential ORI1 inhibition is a potential strategy to prevent ductal damage in acute pancreatitis. So to summarize this part of the talk, we can say that alcohol and bile acids can increase the ORI1 mediated extracellular calcium influx, leading to sustained intracellular calcium elevation, which will inhibit pancreatic ion secretion and fluid secretion that can contribute to the increased severity of acute pancreatitis. On the other hand, when we can inhibit ORI1 mediated calcium influx, we can protect the fluid and bicarbonate secretion of the ductal cells, which can actually lead to decreased severity of acute pancreatitis. In the next series of experiments, we investigated the potential beneficial effect of ORI1 inhibition in chronic pancreatitis. And in our previous publications, we highlighted that in chronic pancreatitis, we can observe decreased intraluminal pH and decreased pancreatic ductal fluid secretion, leading to intraductal obstruction due to the impaired function and impaired expression of CFTR chloride channel in the ductal cells. So first we administered the selective ORI1 inhibitor in a chronic pancreatitis model to mice, and we were able to demonstrate that under these conditions, ORI1 inhibition protects the exocrine pancreatic tissue and decreases the fibrosis in the chronic pancreatitis mouse model. And we were also able to demonstrate that ORI1 inhibition significantly protects CFTR expression on the luminal membrane of pancreatic ductal epithelial cells, and also significantly increases the CFTR-mediated chloride secretion. So, we were able to show that in chronic pancreatitis, sustained calcium elevation due to the increased activity of ORI1 can lead to decreased CFTR expression on the apical plasma membrane. However, when we inhibit ORI1-mediated calcium influx, we can protect the apical expression of CFTR, and we can also increase the pancreatic ductal secretory processes. So to summarize my talk, I would like to highlight that constitutive ORI1 mediated calcium influx maintains CFTR activity under physiological conditions, where high calcium nanodomains are translated to high cyclic AMP nanodomains. On the other hand, under pathophysiological conditions, sustained extracellular calcium influx contributes to ductal cell damage during acute pancreatitis. In addition, ORI1-mediated calcium influx promotes functional damage of the ductal cells in chronic pancreatitis. Therefore, inhibition of ORI1 may have a potential beneficial effect on the ductal cell function in acute pancreatitis and also in chronic pancreatitis. Finally, I would like to acknowledge my colleagues who contributed to the experimental work. I also would like to highlight our collaborators and sponsors, including Calcium Medica, who developed the selective ORI1 calcium channel inhibitors. And I would like to thank you very much for the attention.
Thank you very much indeed, Josef, for this very nice and very interesting presentation.
Thank you very much.
It would appear, nevertheless, that there is a little bit of contradiction here, if I can sort of be a bit provocative here. So on the one hand, your results confirm the sort of general feeling that inhibition of ORI1 channels is a good thing in acute pancreatitis. On the other hand, in general, I think you and Peter Hecke have always maintained that fluid secretion from the ductal cells is an important function and also is an important protection against acute pancreatitis. Now, when you inhibit the ORI1 channels, then you will inhibit the calcium activation of the adrenal cyclase, which in turn will inhibit activation of CFTR and then inhibit fluid secretion. So that would seem to be a bad thing. Could you just comment on this potential discrepancy?
Yes, thank you very much. This is actually really a crucial thing which we should emphasize as much as possible, because this seems to be a contradiction at the first sight, but we actually have data to support that. I was not showing it due to the lack of time, but we have data to support that that the constitutive ORI1 activation, which we can observe in pancreatic ductal cells, is actually like I would say 10–15% of the total activation of ORI1. So it's a really low activity level which is maintained in pancreatic ductal cells, and it really seems to us that this is actually like a basal secretory. This maintains a basal secretory activity, and even if we inhibit ORI1 calcium channel, we can stimulate the CFTR-mediated secretory function with other agonists, like, for example, you can think about secretin, which is acting quite independently from the calcium. So there are ways which still can activate or stimulate the pancreatic ductal secretion, even if the ORI1 calcium channel is inhibited. This is kind of a basal unstimulated secretion, which is governed by the Orion calcium channel. On the other hand, in the acute pancreatitis situation, when you have these toxins present, like alcohol, bile acids, or any other toxins that actually generates a sustained elevation of intracellular calcium, and downstream damage due to that would cause much more damage than like inhibition of Orion channel. So I think it's really a cost-benefit issue, and in acute pancreatitis, as you also showed in many work, a crucial thing is to prevent the sustained elevation of intracellular calcium. So I think you gain much more with that than you lose on the other side.
Okay, thank you very much. I think that is a very good answer and clarifies the situation extremely well. Just a very short question. Have you got any plans to measure cyclic AMP changes in these cells?
Yes, yes, we are working on that. It's not that easy because the yeah. So it's a nanodomain issue, right? So we talk about a couple of hundreds of nanodomains, and we really have to localize the sensors very precisely to the proper place. Because the global measurement is really very similar with calcium, right? That you cannot really measure global calcium because that will be misleading. So we really have to figure out how to introduce targeted sensors in the primary cells which maintain polarity and apical nanodomains. It's a quite tricky question.
Indeed, yes. Well, anyway, good luck. It's a very important investigation that you have embarked on, and I think you made really very impressive progress. We have to move on because we're already a little bit behind time. And so it's a pleasure to introduce Professor Albrecht Schwab from Münster in Germany, who will give us a talk about ion channels and transporters in pancreatic cancer pathophysiology. So Albrecht, the floor is yours.
Dear ladies and gentlemen, thank you very much for giving me the opportunity to present our work at this meeting. And most importantly, I would really express my deep admiration for the organizers, that they still made it possible under these severe conditions of the Ukraine war to have this meeting. So I was asked to give an overview about ion channels and transporters in cancer pathophysiology. And this is kind of a challenge to do this in 50 minutes, but I will do my best. So I would like to introduce you the concept of the transportome. So the transportome means it's the entirety of all cellular ion transport and ion channels and transporters and pumps, so all ion transport proteins. And why do we think that these transport proteins are so important? So when we look at the tumor as a tissue, we can distinguish a cellular component and the extracellular component, the tumor microenvironment. So the cellular components, of course, not only includes the tumor cells, but also all other tumor stromal cells, like in pancreatic cancer, for example, the stellate cells, but also immune cells, endothelial cells, fibroblasts. So, and in all these cells, we have membrane transport proteins located in the plasma membrane. And this is and thereby they can communicate between the intracellular component and the extracellular component of the tumor tissue. And they do this in 3 different ways. So first of all, they do modify the extracellular environment. And this is easily explained with the example of the pH. Protons generated intracellularly are transported out of the cells by transport proteins to cause the extracellular acidification. So transport proteins can also sense the position of the extracellular environment, also the pH, for example, which inhibits a number of ion channels, or ATP, so ligand-gated ion channels that are activated by ATP, purinergic signaling. And finally, many of these transport proteins are central elements of intracellular effector transduction functions, which then control and regulate all the typical functional features, behavioral features of tumor cells and of the tumor stromal cells. So how this works specifically, I will show you with 3 examples, but before that, I would like to point out, of course, so the vision we have, so the ultimate aim we're pursuing is that therapeutic targeting of the tumor transportome has to be also included in the therapeutic repertoire in oncology. So some general considerations before going into detail. Transport proteins may sound as peculiar targets in oncology, but nonetheless, they have been validated and very effective drug targets since decades in other medical disciplines. And 15%, so that's 15% of all prescribed drugs target transport proteins. So, and in terms of oncology, it's also interesting to note that 90% of the tumors are of epithelial origin, and in epithelial cells, the transport proteins are the working horses. So one can say that in tumor cells, these working horses are misused by the carcinoma cells. And importantly, also, we found that essentially in all tumors, transport proteins are expressed in a dysregulated way in all tumor and stroma cells. And they are only active when really needed, so which limits the adverse side effects of these transport proteins when they are therapeutically targeted. So as I said, I will show now 3 examples from our lab, how to illustrate the really important role of transport proteins in oncology. So I will begin with a potassium channel, a calcium activated potassium channel in non-small cell lung cancer. So calcium activated means the channel is activated when the intracellular calcium concentration is rising. So we found in a cohort of stage 1 and stage 2 lung cancer patients that there was that the potassium channel expression had a strong predictive power with respect to the prognosis of patients. And we went a step further and looked why we have this difference in channel expression, and we found that it's the methylation of the channel promoter. And low methylation, which is equivalent to high channel expression, predicts a much poorer prognosis than a high methylation, which corresponds to a low channel expression. So then the question rises, why does increased channel expression cause a poor prognosis for these patients? Well, to give you some answers, what we found is that the blockade of this channel in a xenograft tumor model in mice, we found that blocking the channel inhibits the proliferation of the tumor cells, so that tumor growth here under control condition is strongly retarded when the cells are blocked with this channel, when the channel is blocked with Seneca POC, a small molecule, which is a fairly specific inhibitor of this potassium channel. So proliferation is inhibited in vivo, and we also found now in vitro experiments that inhibiting the channel induces apoptosis. And interestingly enough, here we show this effect in erlotinib-resistant lung cancer cells, NSCLC cells. So interestingly enough, that blocking the channel can also overcome the resistance towards these EGF tyrosine kinase inhibitors. As you can see here, so erlotinib by itself has no effect on apoptosis in these resistant cells, whereas the combination of erlotinib plus potassium channel blocker induces marked apoptosis. Just for comparison, a conventional, so to speak, conventional chemotherapeutic drug, doxorubicin, so it's as strong as with doxorubicin. Okay, next example is the sodium proton exchanger, NHE, and now in pancreatic cancer, PDAC. So before introducing our work on this transporter, we need to have a look back on the pancreas physiology, and in particular on the unique pancreatic pH landscape. So it's well known that the healthy pancreatic ducts secrete an enormous amount of bicarbonate. Up to 150 millimole per liter is the bicarbonate concentration in the pancreatic juice. So by the nature of the chemical origin of bicarbonate, which is the result of the reaction of CO2 and water, catalyzed by the carbonic anhydrase, we always have the equimolar amount of protons to be considered. So that is when 150 millimolar bicarbonate are secreted into the duct, we need to get rid of 150 millimolar of protons. And of course, they are now secreted into the interstitium. So the consequence is that whenever we have bicarbonate secretion, so after a meal, we have an intermittent acidity of the pancreatic stroma. And all cells which are residing in the pancreatic stroma and of the healthy pancreas are exposed to a massive intermittent acidity, and this keeps the stroma quiescent, which we know in the meantime. So pancreatic cells are used to handle huge acid-base fluxes. This is the positive part, but on the other hand, these acid-base fluxes are also a potential threat to cell integrity. So when PDAC now develops, so in the intermediary states, the secretion dwindles down, and then in PDAC we have the full-blown PDAC. We again have the extracellular tumor acidity. So the tumor-induced acidification, but all cells are well prepared to handle this acidity, and this promotes, in our view, cancer invasion and metastasis. So we state this kind of provocative hypothesis so that the pancreatic pH landscape constitutes, so to speak, the red carpet for the rapid tumor cell invasion. So important to know, so the extrusion of protons into the interstitium and the healthy pancreas, but also in the tumorous pancreatic tissue. Of course, we need transport proteins, and one of them is the sodium proton exchanger. And we can go back to this scheme, is so that these transport proteins, sodium proton exchanger, modifies the extracellular environment, so the microenvironment, and this pH microenvironment is also sensed, again, by, among others, the sodium proton exchanger. So what we were interested in is the role of the sodium proton exchanger, so the isoform NHE1, in pancreatic stellate cells, which of course well known as the producers of the fibrosis in pancreatic cancer. So when we isolate pancreatic stellate cells from a healthy pancreas, it looks like this. The activation marker alpha SMA, alpha smooth muscle actin, is not visible, so these cells are in a quiescent state. But this is well-known experience when you isolate stellate cells and put them into culture on the hard plastic dish. So they activate immediately within a couple of days. The cells look like this. They become larger and abundantly express these alpha smooth muscle actin. So this is a measure of quantifying the activation of these cells. So what we now did was to isolate the cells in an acidic environment, so to keep them, the idea was so that the acidity keeps them resting, quiescent, and we kept them for 3 days, and then we changed the medium again to an acidic medium. So they were kept under acidic conditions for 6 days. We know that they are still viable, and this is not a problem for the cells. On the other hand, so the other group of cells, I mean, we also isolated them in the acidic medium, and then exchanged the medium for the normal pH medium, pH 7.4. So what did we see? So in the acidic environment, the cells are quiescent, and they remain quiescent, judged by the alpha SMA expression. However, when relieving the acid break, so to speak, then the cells become activated. And we did a lot of other experiments, did a dose response curve to see these pH effects. And we can see that, I mean, note the logarithmic scale, so that the alpha SMA expression, I mean, drastically rises when the acid pH is exchanged for a more alkaline physiological pH. And this is also reflected by the mechanical properties of the cells. I mean, also on a softer substrate, this is still possible. Now, this was tissue culture plastic, and those were on hydrogels. So the acidity keeps the cells inactive, the stellate cells. So then we tried, so after having the cells being activated, so after relieving the acid break, so we tried to, so we studied what happened when we then put them back to acidity, so like in the tumor. But here we see that then the acidity does not cause an inactivation. Once they have become activated, they remain activated. And this is the statistics, so there was no difference. Okay, so we can conclude from these experiments, so the quiescent stellate cells and the healthy pancreas, they are kept quiescent by the intermittent acidity. So when we look for the tumor condition, so we have the also we start off with a quiescent PSC, but this acidity becomes diminished. This intermittent acidity after a meal becomes diminished, and this causes an alkalinization of the pH values and reprogramming no longer inhibition of the cell cycle, and among others by HIF-1 alpha and YAP-TAZ signaling, the stellate cells become activated. And this is in large part also due to an activation of the sodium proton exchanger. And this in turn then causes fibrosis in PDAC. And we have now first evidence that NHE1 blockade is able to attenuate the fibrosis in pancreatic cancer. Okay, so now the third example is focused on mechanosensitive ion channels and pancreatic cancer. So the pancreatic cancer is also characterized by a fibrotic, as we had talked about already, and stiff extracellular environment, which is in addition under pressure. So we wondered, what is the role of mechanosensitive ion channels in pancreatic stellate cells? Well, what we tested first was whether mechanosensitive ion channels like piezo1, for example, is able to sense the environment of the pancreatic tissue, and thereby we created hydrogels of defined stiffness, also with a gradient of stiffness. So left-hand side is a stiffer gel, and right-hand side is less stiffness. And what we can see is that the cells are steadily moving towards the stiff part of the gel. So they are performing durotaxis. And this is the statistics. So this rose plot clearly shows that the preferential movement direction of the cells was towards the stiffer area of the gel. And when we inhibit mechanosensitive channels, I mean, in this case, it's a spider toxin, which is an unspecific inhibitor of different mechanosensitive ion channels, we see that there is no longer any preferential movement, so that durotaxis in this case has been inhibited. And we also know in the meantime that this is in part also due to piezo activity. So the mechanosensitive channels sense the properties of the tumor microenvironment, in this case of the stellate cells. But they also exert force onto the extracellular matrix. And here we have a spheroid of stellate cells, and the cells are moving out of the spheroid, so like the sun is rising. And what you should focus on is now these little dots in the background, so in the matrix, how they are being pulled towards the emigrating cells. Yeah? So the tumor cells, or the stellate cells, are pulling on the matrix, so they are stiffening, so to speak, the ropes, and only then the cells are able to leave the spheroid. So we quantified the movement. We quantified the movement of these beads. We measured a certain speed with velocity, how these beads were moving. And what's shown here, that when we activate one of these mechanosensitive ion channels, the piezo1 channel, then these cells, this spheroid pulls stronger onto the extracellular matrix. So exerts force onto the extracellular matrix, and thereby modifies the extracellular environment. So what I have shown you is, I hope that you could follow me, so that the transport protein, so the transportome in the plasma membrane, in this case, and the tumor cells, also and the stellate cells, so they're able to modify the extracellular tumor microenvironment. So the pH, for example, also the mechanics. And then these transport proteins are sensors, for mechanics, also for the pH. And finally, many effector functions are also modified by the transport proteins. Proliferation, apoptosis, for example, the potassium channel, the lung cancer cells, migration, durotaxis in case of the stellate cells, and also the mechanics of the stellate cells themselves are also modulated by the mechanosensitive ion channels. So when we put this in a bigger framework, I mean, you may know these famous papers by Hanahan and Weinberg, where they introduced or defined the hallmarks of cancer, so different properties which are found in essentially all tumor cells, all cancers, or cancer cells. And the conventional view is in terms of targeted therapy, so that specific molecules, signaling molecules are addressed, for example, EGFR receptor inhibitors, to target, to inhibit certain aspects of these tumor properties. So this is kind of the conventional oncological view, and what we are now proposing is that all these hallmarks can also be targeted by ion channels and ion transport proteins. And this is, so to speak, our ambition, so that, I mean, this knowledge also now enters into the clinics. And this is very relevant because there are a lot of inhibitors available for these transport proteins. I mean, as I said in the introduction, they are used for different other disciplines, and we just have to repurpose these drugs in oncology. Okay, last but not least, a disclaimer, so to speak. So our work on pancreatic cancer has been funded, among others, by 2 European networks. So the former one was IonTrack until 2015, and the current one is Phyonic, which specifically is on pH and ion transport in pancreatic cancer. And with that, I would like to close and thank you for your attention, and thank to my team and also to collaborators within the Phyonic team and Carsten Müller-Tidu for the lung cancer project. Thank you very much for your attention.
Well, thank you very much indeed, Albrecht, for that very comprehensive and very innovative overview, pointing out so much about ion channels and their role in cancer and all the potential there is for the treatment. There are a number of questions that have already been posed, including whether or not, this is from Alexei Tepkin, are calcium-sensitive potassium channels relevant to other types of cancers?
Yes, indeed. So thank you very much for your comments. And so this calcium-sensitive potassium channel is actually one of the best studied channels in cancer. So there are many examples where the channel has been found to be upregulated in cancer tissue, and it's known to regulate proliferation, migration, apoptosis, and in many different cancer types. And what I like very much about this channel, so there's a drug which there's a fairly specific inhibitor for this channel, which had already been in phase 3 clinical trials for sickle cell anemia. So, and it was quite well tolerated. It didn't make it in sickle cell anemia, but I mean, this is our hope that, I mean, there was also another question in the chat, I think it was from you, so where to start therapeutically. I mean, so there are many drugs available which are already used clinically, or which had at least gone to phase 3 clinical trials, sometimes big phase 3 clinical trials. So one of them was this potassium channel blocker, Seneca-POC, and also the NHE1 inhibitor we were using, Keriparide, was used for cardiac reperfusion ischemia therapy. The clinical trial wasn't really that successful, but it was a huge trial. And this would be another drug which could, I mean, revitalize, so to speak, for different indication. And what people, and including our group, are now doing is to use genetic cancer models, also PDAC models, and start like a therapeutic trial with these channel and transporter blockers as a supplementary treatment modality in combination with conventional chemotherapy.
No, that's really, really exciting. There's one more question that we've got time for, because I think that we're covering the coffee break at the moment till 11. Do you have, this is from Fred Gorlick, who's one of my co-chairs. Oli has had to go off for another meeting. Do you have thoughts on what pancreatic stellate cells sense when extracellular pH is changed to regulate activation, e.g. cell surface proton sensors, intracellular proton sensors, kinases, or general responses?
So, I mean, this is a very good question, and of course, the most evident way would to sense via our, I mean, at least through our eyes, is to sense it via the transport proteins, and the extracellular pH is usually also transmitted to a change of the intracellular pH. And what we've found so far is that YAP-TAT signaling is also regulated, for example, by the NHE1 transporter. So when it's inhibited, we see a change of the YAP-TAT's translocation to the nuclei. So in essence, in our view, it's probably more the intracellular pH which matters. But of course, there are also direct proton sensors, so G-protein coupled proton sensors, which are also expressed in the stellate cells, but we have not yet addressed their function.
Okay, well, Albrecht, thank you so much for a great presentation and for addressing the questions. There may well be more in the chat that appear, and I'm sure other people will be keen to communicate with you. I would like to thank, on behalf of my co-chairs and myself, the speakers for making such great presentations, such an interesting area, really potentially of really great impact to patients worldwide. I'd like to thank Fred for staying the course throughout the middle of the night, so I hope you can get some rest and sleep. And I'd particularly like to thank Ukraine organizers for their sterling endeavors faced with what they have to put up with. As Oli said, our thoughts are with you. And he's put on the post an article he's written in Function, the journal Function, about what scientists should do in times of war. So leave you all with that message and goodwill to all of you.